Vamos a celebrar el último coloquio de este ciclo del ciclo 2018-2019 y paso la palabra a la vicepresidenta de Relaciones Internacionales, doña Elena Domínguez.
Buenos días a todos, o buenas tardes a todos y a todas, y bienvenidos, queridos colegas, de nuevo a esta casa que, como se ha dicho ya en repetidas veces, por supuesto, es la vuestra. Se ha anticipado, es decir, mi nombre es Elena Domínguez, soy vicepresidenta de Relaciones Internacionales, y lo primero que tengo que hacer es excusar la ausencia de la presidenta, de Rosa Menéndez, porque su agenda es terrible, básicamente, y no le ha permitido estar hoy aquí. Me consta y soy consciente del interés que ella tenía, porque participó en uno de los proyectos, del interés que hoy tenía en asistir a esta última conferencia, a este último coloquio Marie Curie, pero le ha sido totalmente imposible. Y aquí estoy yo, dándoos la bienvenida, y si me voy a permitir la arrogancia de un pequeño cierre de este extraordinario ciclo. Quiero, en primer lugar, sin duda, agradecer, y es honor y es deber, agradecer a los organizadores de este coloquio y, en particular, a José Miguel Mancheño, del Instituto de Química Física Rocasolano, a Sagrario Martínez, del Instituto de Estructura de la Materia, Rebeca Denalda, del Instituto de Química Física Rocasolano, Javier Portilla, del Instituto de Óptica, y a Carlos Sabín, del Instituto de Física Fundamental, por este largo e intenso esfuerzo que habéis desarrollado en organizar estas conferencias y estos coloquios. Y me voy a permitir, por el hecho de ser, si digo esto delante de la presidenta, por ser externa a esta casa, en el momento en el que vosotros iniciasteis vuestro primer coloquio, en octubre de 2017, me voy a permitir hacer un balance global desde como parte ajena, viéndoos lo que habéis hecho y la huella que habéis dejado en la web, con vuestros pósters, con vuestros vídeos, la huella que habéis dejado. Me voy a permitir hacer un assessment, un balance global de este hecho. Y el balance global de este hecho es óptimo, es buenísimo, es extraordinario. Y os voy a decir por qué y lo voy a justificar. No simplemente porque os habéis esforzado, sois unos investigadores de pro, los asistentes aquí habéis atendido a unas extraordinarias conferencias, pero además porque estáis, y si me permitís decir, estáis dentro y completamente de la línea estratégica, científica y tecnológica que desarrolla la actual presidencia del CSIC. Y yo la voy a resumir aquí en la combinación de unas cuantas IES para dar lugar a una I mayúscula. Vamos a ver. En primer lugar, todos estos coloquios, los 8 que habéis celebrado hasta ahora, e incluido el 9º, son un ejemplo maravilloso de interdisciplinaridad. Algo que a los científicos, como sabemos mucho de muy poco, algo que a los científicos nos cuesta en principio y a priori algo asumir. Es decir, pero los coloquios en general, las conferencias, han sido un ejemplo maravilloso por ejemplo, de cómo la física está contribuyendo de una forma espectacular a la biología. O cómo, por ejemplo, muchos ejemplos, fundamentalmente han sido en este caso, a tenor del comité organizador de estos coloquios, os habéis centrado en la materia, en la física, en esta tecnología, para ver cómo incide en cualquier rama del saber. Luego, la interdisciplinaridad ha estado garantizada. La segunda I nos viene de, casi ça va de soi, ¿no? Es decir, la I es de internacional. ¿Por qué? Porque la ciencia es internacional, pero no por el hecho de que la ciencia sea internacional, la internacionalización de este coloquio es espontánea. No, habéis hecho un esfuerzo en traer a personalidades, en traer a científicos de cualquier país del mundo, traerlos aquí para que nos enseñen cuáles son sus puntos de vista, sus tecnologías, sus técnicas y sus perspectivas en este coloquio. Luego, habéis hecho también un esfuerzo de traer a personas del ámbito externo a nuestro país a estos coloquios. La tercera I es mucho más difícil, extraordinariamente difícil. Es en la que, la Comisión Europea lleva no sé cuántos programas marco persiguiéndola denodadamente, ya no sabe qué inventar para que los científicos, para que la investigación sea intersectorial. Aquí viene la dificultad, es decir, cómo mezclar, cómo combinar, cómo interfaciar lo privado con lo público, cómo interfaciar la investigación y el desarrollo del conocimiento con la industria, con la empresa, cómo interfaciar nuestro conocimiento, nuestra tarea diaria con la sociedad. Y no solo a través de la divulgación, no solo, sino además con un contacto más permanente de educación, de difusión, etcétera, etcétera. Luego, este aspecto intersectorial, que es un reto permanente en lo que es toda la actividad y toda la política científica, por supuesto vosotros también lo habéis cumplido, porque originariamente, y doy fe de que no he hablado con ninguno de vosotros, sino que lo he percibido del testimonio, de la huella que estáis dejando en Internet, todas las comunicaciones y el origen de estos coloquios es comunicar a la sociedad, difundir vuestro conocimiento para hacerlo más accesible. Y esta es nuestra obligación y esta es también nuestra responsabilidad. Luego, este aspecto, estos coloquios, en esta miscibilidad con la sociedad, también lo habéis cumplido y, desde luego, enhorabuena por ello. Y me voy a permitir una cuarta I, me voy a permitir otra más, antes de llegar a la I capital, a la I mayúscula. Me voy a permitir otra, que es la de la inclusión. Y aquí las mujeres de inclusión sabemos mucho. Y sabemos mucho de mujeres porque la primera vez que yo tuve que hablar de inclusión y me dijeron que el género estaba incluido en la inclusión, me negué rotundamente a incluir el género dentro de la inclusión. Creo que la sociedad debe de estar mucho más avanzada como para tener que meter los aspectos de género en la inclusión. A nada que uno penetra en la inclusión, teóricamente, a nada que uno penetra se da cuenta de que el género también hay que incluirlo en la inclusión. Muy a mi pesar, porque es cierto, la visibilidad que tenemos las mujeres científicas, las mujeres en general, conforme progresa la capacidad en la toma de decisiones, sabemos que proporcionalmente al aumento en la competencia y en la capacidad para la toma de decisiones, disminuye nuestra presencia y disminuye, por tanto, nuestra visibilidad. Vosotros lo habéis hecho en estos coloquios extraordinariamente, porque habéis desarrollado un equilibrio perfecto de conferenciantes y lo habéis hecho, me imagino que no sé si espontáneamente o deliberadamente, pero a la hora de ver las cifras, haber tenido a 5 ponentes del género masculino y a 4 ponentes del género femenino no es, por lo general, habitual, sobre todo en estos temas que rozan con la física, y muchas de las ponencias han tenido que ver con la física. Luego, enhorabuena por todas estas IES que habéis cubierto, porque este es el único camino que, desarrollando una buena ecuación, una buena función para combinar todas estas IES, podremos llegar a la innovación. Y solo así, la innovación no es espontánea, la innovación, hombre, todos sabemos lo que puede ser la serendipity, pero en definitiva, la innovación, esa innovación que penetra, esa innovación que nos hace crecer socioeconómicamente, es largo, difícil de obtener y requiere de todo este entramado, de todas estas IES. Luego, enhorabuena en estos coloquios, que sin duda son unas semillas, una contribución muy importante, y os diré, a la política científica que ahora mismo la actual presidencia del CSIC está desarrollando. Y no en balde os puedo decir que con más énfasis en unos aspectos, la vicepresidencia de Investigación Científica y Tecnológica, liderada por Jesús Marco, y la mía propia de Internacionalización, nos combinamos extraordinariamente para hacer ecuaciones, para hacer funciones que resulten en una innovación, que resulten en un conocimiento y en un desarrollo científico que mejore nuestra sociedad. Y todas vuestras charlas, además, y todas vuestras conferencias tienen, además, un valor extraordinario. Es decir, habéis tocado una serie de temas y me permito decir, habéis incluido desde los telómeros y su papel en el cáncer y en el envejecimiento. Habéis traído aquí a Margarita Salas, que nos ha contado su experiencia de vida, Y Margarita Salas, si no lo negamos, decíamos, y no lo podemos negar, es un referente en el Consejo Superior de Investigaciones Científicas, lo ha sido, lo es y lo seguirá siendo. Habéis traído también aquí a expertos en cómo ver lo pequeño, lo más pequeño, con rayos X, en pulsos de femtosegundos, etcétera, etcétera. Y hoy vamos a ver, y no voy a estropear el comienzo de nuestra maravillosa película, de nuestra maravillosa aventura hoy, y hoy vamos a ver algo fascinante. Vamos a ver cómo podemos entender mejor a una célula, cómo esa célula cambia la dinámica de esa célula en unas dimensiones espaciotemporales que son moleculares, es decir, vamos a ver ojos moleculares para interpretar, en definitiva, qué es lo que perseguimos, para interpretar cómo es la vida, para entendernos al nivel más elemental de nuestros sistemas vivos, como es, en este caso, la célula. Luego, no me queda más que, de nuevo, felicitaros por estos coloquios. Cerráis hoy un ciclo, enhorabuena. Yo solamente deseo que esto sea el comienzo de otros nuevos ciclos, que seguro que tenéis imaginación para diseñarlos de nuevo. Marie Curie siempre es un nombre que nos inspira a todos para hacer actividades. Podéis, sin duda, contar con el actual equipo directivo del CSIC para este tipo de iniciativas o para otras equivalentes. No lo dudéis, cualquiera de las vicepresidencias os podrá ayudar, y no que decir la vicepresidencia adjunta de Cultura Científica, que seguro con la que también estáis colaborando. Luego, bienvenidos de nuevo a esta casa. Yo creo que hoy es un día de felicitación para todos y, desde luego, nos espera un programa y una hora espectacular. Y a la doctora María García Parajo la van a presentar a continuación y ya veréis las cosas tan fascinantes que puede hacer. Muchísimas gracias a todos vosotros por vuestra atención.
Bueno, primero quiero agradecer a la vicepresidenta las palabras tan cariñosas que nos ha brindado a estos coloquios y nos congratulamos del apoyo institucional que siguen manteniendo estos coloquios, apoyo que siempre hemos tenido, pero que explícitamente hoy se nos ha reconocido. Has hecho un resumen de los ciclos que no lo podríamos haber hecho mejor nosotros, así que muchas gracias por estas primeras palabras. Y ya paso a presentar a nuestra conferenciante de hoy, a la doctora María García Parajo. Tuvo la licenciatura en Ciencias Físicas en el Imperial College. A continuación hizo el doctorado en Física Electrónica bajo la supervisión del profesor Mino Green. En el año 1996 obtuvo un postdoctorado en el Instituto de Tecnología en la Universidad de Twente en Holanda. A continuación obtuvo otra beca en el Royal Netherlands Academy. En el 2002 consiguió una plaza de assistant profesor también en el Instituto de Nanotecnología y finalmente en el 2004 llegó a ser profesor asociado en la misma institución. En el 2005 volvió a España y volvió gracias a que pudo ser líder en el grupo de nanobiofotónica del Instituto de Bioingeniería en Cataluña, en Barcelona, y actualmente es la jefa del grupo del Single Molecule Biophotonics en el ICFO, en el Instituto de Ciencias Fotónicas de Barcelona, donde actualmente es profesor de investigación y CREA. Su investigación se ha centrado en el desarrollo de técnicas ópticas avanzadas aplicadas al estudio de los procesos biológicos a nivel del single molecular en organismos vivos, llevando los conocimientos biofísicos hasta las cuestiones biológicas fundamentales orientadas hacia la salud y las enfermedades, incluyendo la biología molecular y la inmunología. Sus trabajos se han publicado en más de 100 artículos, en más de 110 artículos y han sido citados más de 4.500 veces, con lo cual la relación es una relación que hace que tenga un índice H de 43. También, gracias a estos enormes trabajos que ha realizado y que ha conseguido publicar, ha sido también miembro de diferentes organizaciones relacionadas todas con la biotecnología, y destacar pues, que ha pertenecido al Comité Ejecutivo de la Sociedad Española de Biofísica. Darte las gracias nuevamente por haber accedido a participar dentro de estos coloquios y ya sin más, pues te doy la palabra para que nos presentes la charla.
Vale, pues buenos días a todos y muchísimas gracias por la invitación. La verdad es que es un poco imponente toda la charla, todo el protocolo. No estoy acostumbrada básicamente a tanto protocolo. La charla me ha parecido estupenda, inspiradora y me parece espectacular tener gente a nivel político de toma de decisiones que esté de alguna manera tan compatibilizado realmente con la ciencia y, como me acaba de decir ella, que realmente se lo cree, y eso se nota por la introducción que has hecho. Entonces, muchísimas gracias de nuevo y comienzo. Lo primero que voy a hacer es pediros disculpas. Voy a dar la charla en inglés, pero obviamente el castellano es mi lengua materna. Me podéis preguntar todas las preguntas que tengáis en castellano, pero me siento mucho más cómoda si doy la charla en inglés, para no liarla y terminar con un Span-English de vergüenza. So I'll just switch immediately. So as it was said, I work in Barcelona in the Institute of Photonic Science since 2011. And actually I have been abroad for many years. I came back to Spain in 2005. I got an ICFO position, and I was first in an institute in bioengineering, and since 2011 in the Institute of Photonic Sciences. And so what I want to tell you today, and I got instructions to give a very general talk, so again, I apologize if somebody wants to have more technical details or more insight in terms of the biology. I will be happy to discuss that afterwards. But in principle, it's a sort of a general talk with the idea that at least you get a glance and you get an idea of what is going on in the field. And I say this like new eyes in order to look at the nano-world of living cells. And what you see in here immediately, this is not from my group. This is already you can actually download it on the internet. What you see in here is just by using light and by using fluorescence microscopy, you can essentially label particular components in the cell, and nowadays you can even see processes alive. So you can see dynamics on how cells move, how cells interact, and for instance, in this particular case, the process of cell division in there. So fluorescence microscopy is one of the major tools for biologists in there. And so let me just give you a first idea, so historically, on how the world of light microscopy essentially came about. And probably the first useful microscope, it was invented by Antonie van Leeuwenhoek, Dutch person, in 1679. And this is essentially the microscope that he came with. And what you see in here is essentially the lens. So actually the microscope, this magnifying lens, is essentially here. And all this part that you have in there is essentially to move the specimen very close to the lens so that you can get the magnification that you see in there. And so the core component is this one. And using this microscope, Antonie van Leeuwenhoek was able to see details of already this organism, which you can imagine, but at this time, you will have to draw it by hand, okay? And so already at that time, and this is actually a curiosity, van Leeuwenhoek, he essentially said this. I said, I do not think that mankind would ever advance that far to watch the part of the water, to be able to watch molecules in that. And one of the things that actually happened is that van Leeuwenhoek was wrong. In 1993, actually individual molecules became available. So in 1993, it took hundreds of years, but we were able in 1993 to be able to detect the fluorescence from individual molecules. And this actually has been a breakthrough to all the modern fluorescence microscopy that we know nowadays. Now, at that time, we were able to essentially see the fluorescence that it was emitting each individual molecule, but soon after that, we actually started to find out that those fluorescent molecules also had different properties. So they could emit light at different wavelengths, and therefore having different colors, but also individual molecules, they have a defined dipolar orientation. And that means that we could follow where the molecules were oriented in three-dimensional conditions. And so light brings you that property, fluorescence gives you that property in there. And that was actually in 1993. Another anecdote in here, and I just want to test a very simple test in there, is that those eyes to look into the biology or to look into organisms using microscopy, we can actually use also different eyes by looking at different spatial scales. And I would like to ask you whether any of you is able to recognize this image in there. Does anybody knows what is this? This is, yes, indeed. So this is the old light, so it's the Hubble Deep Field. So what you see in here is the sort of the far-reaching light from the universe. And all these spots that you see in here are galaxies. They are not, you are not looking at galaxy. What you're looking in here is the interaction of the light with a galaxy. And this is what you see in these ones. Now I'm going to put next to you an image of individual molecules. Doesn't look very similar. And so essentially what you're looking in here is a spatial scale which goes into 10 to the 26 meters, which is our universe, and we're looking at 10 to the minus 9 nanometer scale, which are these molecules in there. These, obviously, we obtained these images with the Hubble, with the telescope. These were obtained using very fancy microscope. But it's coincidental that you see that the features are the same. In here, you obtain interaction of light with a galaxy. In here, you obtain interaction of the light with the fluorescence from a molecule. So now let me just tell you a little bit about how it did progress. So it took another 200 years until microscopes, they became quite sophisticated. So now you look at this one, these are the lens in here, that we can have the lens in there, we can have our specimens, and then we can look through that. And actually Fleming was the first one of the first ones to actually use this microscope in there. And again, he was the first one to then look with a higher resolution and then see how cells divide in there. So the entire process of the mitosis. And again, at that time, he had to draw it by hand. Now, I'll show you the image at the very beginning. This is a standard microscope, the ones that we use nowadays. So this is a photograph. So this is a microscope, a commercial one from Nikon from 2015. And this is how much as we have advanced in there. So not only we get this picture directly into this camera, but we can see multiple colors. So where do these colors come from? This is a cell, so this is the same process of cell division in here, but molecular biologists, and this is very important and it goes to the point that you were trying to make, physicists, molecular biologists, chemists, they actually work together to design individual molecules or markers, fluorescent markers, that were specific for different organelles in here, for microtubules, for the nucleus, for the centrosomes, for the cell membrane, and by using the combination of different excitation wavelengths, we were able, and biologists are able to look at these features with much higher resolution, but also with much higher detail. However, there is a problem with fluorescence microscopy or with optical microscopy in general. And so the problems I will just try to illustrate you in here. If you look, for instance, so that will be a conventional, modern, state-of-the-art fluorescence microscopy of different cells in there. So you can see the different cells in here. You can see the actin being labeled in red. You can see the microtubules being labeled in green. And you see the nucleus being labeled in blue. It doesn't really matter, the ones that are not biologists in here, but they're just different organelles. So we have a specificity to label those individual components on individual cells in there. However, we do not see the part of the molecules that compose the cells. We see an average image in there. We cannot see individual actin filaments. We do not see individual DNA that we know that is inside the nucleus. And so if you try to zoom in to gain resolution, to be able to look at those molecules in here, this is essentially what you start seeing. You start seeing that if I try to zoom in, my image becomes very blurry, right? On a scale, so this is 20 microns. If I now try to zoom in even more to a spatial scale in which I should be able to see the sizes of those molecules, you see that my image is completely gone. It's completely blurred. And so the question is, why does that happen? And actually, this is a very old phenomenon. It was already, postulated in 18,000 by Abbe. Is a problem of the diffraction limit of light. And this diffraction limit of light essentially doesn't give you enough resolution to see the smaller part of the component. And I just put this slide in here to give you an idea, so a cartoon for those of you who are not familiar of what does it mean, the diffraction limit of light. And essentially, the diffraction limit of light, you can see it in two ways. So as it was described by Abbe, and this is the diffraction pattern, if you have a lens and you use the lens to image an object, so imagine that you have this very small object in here, and you essentially illuminate that object in order to image it, the light from the object is going to be radiated in an isotropic way. So it goes in all directions. And the lens now has to capture all the frequency components of the light being emitted, which it cannot do. So it essentially only collects a part of that spectrum of emission of the light. And essentially, what happens in here is that when you look at your imaging object, instead of having that very small spot of light, what you essentially get is a distribution with a width that is essentially much larger than the point that you are initially imaging. And this is the diffraction limit of light. So the diffraction limit of light, in another way, which is probably more intuitive, light has a wavelength. In the visible light, it will have a wavelength of about 600 nanometers. And so essentially means that you can never focus the light to a smaller dimension than 600 nanometers. And seen in another different way, if you would have, and so this is also defined, it's the same thing, but it's a different way of defining what the diffraction limit of light is. It's following this Rayleigh criteria. If you would have two different sources of emission of the light, if these two sources are separated by a large distance, the emission of the light from this source is going to be this one. And the emission of this source of light is going to be this one. As long as they are separated, I can distinguish them apart. But I start getting these two objects close to each other, you start seeing that now the emission of one source gets convolved with the emission of the other source. This doesn't come down. And if I keep on increasing this, you will see that for distances at about 250 nanometers, the diffraction limit of light, I cannot distinguish, I get a blurred image because I essentially cannot distinguish these two features apart in there. And so this is what it is, the diffraction limit of light. And it's essentially defined by this equation. So this gives me the maximum resolution that I can get using light, which is the wavelength of the illuminating light divided by 2 times the numerical aperture of the lens that I'm using in here. And in practice, this goes between 300 to 500 nanometers for you to give you an idea. Okay, and so the challenge that we have in here is the following. We can use a standard microscope, a conventional fluorescence microscope, with a resolution that is going to be larger than these 300 nanometers, because this is the diffraction limit of light, and I can use it to see large things, right? Now, if I'm interested in looking at individual proteins, which will have a size of about 5 to 10 nanometers, I need to have a resolution that is around of the same order. And so this is what super resolution microscopy is. This is the challenge. How can we convert this conventional fluorescence microscopy? How can we overcome the diffraction limit of light into something that is going to give us a super resolution? Okay? And this, I can tell you that since the postulation of this fundamental limit of light that still exists, because it's a fundamental limit of the diffraction of light, and that was in 1840, it hasn't been much progress in fluorescence microscopy or in optical microscopy, just because this has been a fundamental barrier. Okay? However, there were some ideas already being put forward, and that led to one of the super resolution techniques. And so one way of breaking this diffraction limit, I told you that this diffraction limit essentially comes from the fact that we use lenses to focus the light. And I can never focus the light to a spot that is smaller than diffraction limit of light. And now, this will be essentially what we also call, this is called far field, so the propagating light, in which I told you that this is my diffraction limit. So if I have a point source in here, I can never focus it to a smaller point in there, which is, again, this lambda over 2 times the NA of light. Now, in 1928, Sir Singer in London, he came up with this very clever idea. And so what he said, like, well, one of the ways to go around with it is that why don't we use a sort of a geometrical confinement? So if we are able to put a plate in here, an opaque plate, let's say a metal plate, that will block all the light, and we make a very small hole in there, and I'll focus the light, so I'll come to this plate with this very small hole in here, what is going to happen is that the light, as I told you, the light is not going to be able to propagate, because light needs to propagate over the dimensions of the wavelength of light. So that is the condition. And so here light cannot propagate, but there is an evanescent field. So there is another evanescent field which is not propagating, but it remains very, very close bound to the size of the aperture, which essentially is able to give me a much higher resolution. And the spatial resolution that it can give me is essentially of the same time, of the same amount as the size of this hole in here. And this is called the geometrical confinement. One of the requirements in there is that, of course, if I have the light, so imagine that I have the light in here, so you see the light in my hand, because the light is propagating through the air, right? If I reduce the dimensions in here, the light is not going to be propagating, but then if I put myself extremely close to where my source of illumination is, I'm able to pick up electromagnetic fields in the near field that give me that high resolution, okay? So that essentially required to make these very small apertures and to place my sample extremely close to it. Now, Singer discussed this idea with Singer already in Singer with Albert Einstein. He wrote a note, he wrote a letter, and Einstein said that was crap, that could never be done, that not existed, that you could not have evanescent fields, and even if you would have, how could you position a sample with nanometric separation between this hole? And how could you make this hole so small? There was no way. And so Singer never published this paper. So that remains, and you can see it's in the National British, in the British Library, you can see the notes that he wrote to Einstein. Now, it took, again, many years, but in 1984, with the advent of scanning probe microscopies, Dieter Paul in the 88 came out with this idea. So he took back the idea of Sinju, because at that time, what was possible, it was to make piezo elements that could position in your sample with respect to a probe in very close proximity and very accurately in the order of nanometers. And the other idea was that Dieter Paul comes with this idea of using an optical fiber. So you use an optical fiber in which light propagates, and then at the very end, you sharpen that optical fiber. And then you cover it with aluminum so that you have a very small spot of illumination in there. If now you put your sample in a piezoelectric element that will bring the sample very close to this sample, then you could have super resolution microscopy. And it took, again, several years to implement this, but this is in the way that it works. So you make these very small probes with a very small hole in here, and that is the cartoon illuminating, and now you scan it in very close proximity to the sample. And every time that the probe, your nano source of light, interacts with your molecule, you would see emission of light. And this is what we did for many years in my lab. So what you see in here is that by using this technique, we scanned over cell surfaces. What you see in here is one of those images. The gray features that you see underneath is the topography of a dendritic cell. And what you see on top are small nanoclusters and individual molecules on the cell membrane of a fixed cell. And so super resolution was already achieved by using this technique. And in that particular time, we obtained a resolution that was in the order of 70 nanometers, which was fantastic already. But 70 nanometers is still far from getting into the 10 nanometers that we wanted to get. And so then my group started to work on a different concept, which is a concept that is based on antennas. The same antennas that we have, that we know. So what does an antenna do? Essentially what an antenna does is that you have the electromagnetic radiation, so the radio waves, and essentially what an antenna will do is that it will interface that electromagnetic far-field radiation with your receiver, right? Your television, your radio. And the antenna, what it's going to do is that essentially it's going to convert this far-field electromagnetic radiation into near-field, that if you put your receiver very close to it, it's going to do two things. One of them is that it's going to amplify the electromagnetic field, and this is what antennas do. That's why we use antennas, to amplify the signal. This is one of the things. But the other thing that it does is that it localizes the electromagnetic field to the dimensions of the antennas. Now, there is a major difference, and that is, of course, if we use radio waves, we need to have these huge, large antennas. If you use for the television, you need to have the typical antennas. If we want to have a similar effect of enhancing the light and localizing the light, we need to fabricate these antennas in the same scale as the wavelength of light. So we need to go and we need to fabricate antennas at the nanometer scale. And so for this, we use techniques in our institute, which are nanofabrication techniques. And so here are some examples and some analogies of the antennas that exist. So everybody knows the monopole antennas, the ones that you put on your car. And so this monopole antenna, essentially what it does is that at the end of the antenna, it picks up all the electromagnetic wave and essentially concentrates, enhances that field, and it sends it to your receiver. Now, the equivalent at the nanometer scale of a photonic nanoantenna is this small needle in there. And what you can see in here is that these dimensions are in the order of 20 nanometers. So we use a specialized fabrication tools to essentially fabricate this very small needle. The length of the needle is about 80 nanometers, will be the equivalent in there. And the confinement of the light, the localization, is at the very end, that part, which it will be 20 nanometers. This is another example, so that is the typical bow tie antennas. So those bow tie antennas do not exist anymore, but those were the ones that I remembered my mom in the televisions, in the earlier televisions, you will have to put that next to the television, and you will have to move it all the time so that, you know, to see the soap operas, the series, because the things were really lousy, right? And so, but this is a bow tie antenna, and what you can see in here is that we're also able to fabricate the similar bow tie antenna now with a gap region, and here is where the field is being concentrated in this part in there, in the order of 30 to 50 nanometers. And this is the equivalent to the antennas that we know, the most normal antennas. They're called Jagihuda antennas because it was this guy, Jagihuda, who invented these antennas. And this is the equivalent of the Jagihuda antenna. And these Jagihuda antennas, essentially we use it, so with the same, so we have the different parts, the reflector part, the feed part, and the directional part. And essentially what these antennas do is that they are directional. So we use them because not only they are able to collect the electromagnetic field, but they also add directionality, and therefore they enhance the collection of the field. And this was also work that we did demonstrating that we can add directionality to individual single quantum emitters. For the purpose, so these are some types of antennas. So the question is, how can we use these antennas to do some imaging? And so let me show you what we did. And so as a demonstration in here, what you see in this image, in there, is a confocal image. So a standard state-of-the-art standard fluorescence confocal imaging of two different molecules. One of the molecules emit in red, the other molecules emit in green. But because of diffraction and the high density of these molecules, essentially what you see at this scale is that everything is blurred, and you see a lot of yellowish in there because it's very difficult to recognize, because of the diffraction limit, what are those individual molecules. If now, instead of illuminating with my objective, I'll come with my photonic antennas on top, on the same part of the sample, this is essentially what we see. So now we see those individual molecules very well separated and in all different colors. And the antenna that we have used in here is what we called a hybrid antenna. So we essentially fabricate this little monopole on top of this bow tie antenna in there. And so if you essentially now go and measure what is the real size in here, this is essentially what we see. So we see that those spots, they have a size of 20 nanometers. So we now have gone for 300 or 400 nanometers diffraction limit down to 20 nanometers in both colors, in the red and for the green molecules. And something that is probably more interesting and extremely revolutionary in here is that not only we get the sizes of about 20 nanometers, but if you look at the center of where my molecule is, I can localize where my molecule is with an angstrom precision, with 2 angstroms precision. And that actually brings the field of molecular biology directly visualizing molecular biology using this type of approaches, because we go in terms of localizations to the angstrom scale. Okay, so I told you about this technique, near field. So this is the use of the antennas in here. They do not rely on lenses. They use the confinement of the light, but it has the drawback that it's a surface-limited technique because I have an evanescent field. So the light decays very fast as I get away from the source, right? Which is this one that I told you. Now, what happened actually in 2006, between 2005 and 2007, is that several papers came out by taking advantage of the properties of individual fluorescent molecules in combination with conventional optics to somehow overcome the diffraction limit of light. And these are 2 of those techniques in here. So one of the things that is important, at least for me as a physicist, is that we do not break the diffraction limit of light. This technique does break the diffraction limit of light because it uses a near-field approach. These techniques do not break the diffraction limit of light, but overcome somehow by using tricks that we can see smaller. All right? And one of these techniques is called a stimulated emission depletion. I know that in the centers of the CSIC, some of the centers of the CSIC have this technique available, commercially available. And in here, the trick is essentially to superimpose two different wavelengths so that on this donut part, we are going to be depleting the fluorescence from many of the molecules in there, so that my spot of illumination essentially reduces. So this is what it is called also PSF engineering, and I won't give you any technical details unless you are interested in that. And the other techniques that are based on the single molecule localization methods. So let me tell you a little bit about this. In 2014, these techniques received the Nobel Prize in Chemistry. And actually, you see in here the lateral resolution that one can typically obtain with the different techniques. So in the case of this technique, which is called STET, or in the case of this technique, which is called ENSOM, we get resolutions which are typically in the order of 30 to 50 nanometers. These numbers have been improving. If I use the single molecule localization method, the equivalent resolution that we're going to get is between 10 to 15 nanometers, and these are realistic values. But the techniques, and this is still and continues to be a challenge, you need to use fixed samples. So you cannot look at living cells, you cannot look at dynamics just because the techniques still are extremely slow. Now, in the case of the STET, I'll give you an example of how does it look like. So now these are individual receptors, so proteins on the cell surface, and essentially you see all different spots in here, and if one quantifies the sizes of these spots, we get essentially a resolution which is in the order of about 100 nanometers. This is in realistic conditions. So that will be the resolution that you will get with a typical STET. Now, if we think in terms of the single molecule localization method, I need to explain to you a little bit the essence of those methods so that you understand this. So I told you that we are able nowadays, in a quite routine way, to detect individual molecules. So we use highly sensitive cameras, very optimized ways of illumination, but we can detect the emission of individual molecules. And so we can make an image of those individual molecules that will be in there, in which each spot corresponds to the emission of an individual molecule. Now, if you project this, so that you also take into account the intensity, this is what I showed you before. This intensity will have a peak like this, and these ones, and so you have these multiple peaks. Now, one of the things that you realize is that the sizes of those spots, again, are diffraction limited. So they have a size of 300 nanometers. But because I have the intensity, if I have those molecules very well separated in space, I can actually pinpoint the center of mass position of that molecule with a much higher localization accuracy. That means I can set where my molecule is with a precision between 10 to 30 nanometers. Okay? And this is the essence of this single molecule super resolution method. So the problem in here is that I can do this as long as my molecules are very well separated in space. But if I'm labeling a sample in which I have a lot of molecules in there, those molecules are going to be all together. So I cannot separate them. And so the trick to super resolution has been to switch off most of the molecules so that only at a given moment a set of molecules is visible. And then do this, and this is what it is illustrated in here. So I have a sample in there in which I have an enormous amount of fluorescence. What I'm going to do is that I'm going to switch all the molecules off, And then I'm going to allow only a few of the molecules to come up. Every time that a molecule comes up, I can determine with very high accuracy, with nanometer accuracy, where my molecule is. And I record that point. And in the next frame, I record multiple other points. And I do that for thousands of frames until at the end, I get a reconstructed image with the molecular positions of where my molecules were in a high density sample. And this method was the one that essentially got the 2014 Nobel Prize in chemistry. And I said in chemistry and not in physics, because the key thing in here is how do you switch molecules on and off, which is a chemistry trick. It doesn't have anything to do with physics. The diffraction limit of light is still there. This is a trick to position with much higher accuracy where my molecules are, but taking advantage of the chemistry of the molecules in there. So what do we get in here? So this is an image of microtubules now. You see the diffraction limit. This is a standard, again, fluorescent image. And now I do the process of switching on and off. And this is essentially the super resolution image that you get on similar structures. And of course, the equivalent resolution that you get, it is just astonished as to compare to what we will get with a conventional microscopy. Now, just I have to speed up a little bit in here. As you can see in here, the images that I have shown you so far are essentially in one color, the super resolution image. Because these techniques can work, but can work with only one color. And one challenge in the field, there are actually two current challenges in the field. First of all, how can I put more colors into that? Because in biology, we want to see interactions between different components, so I want to use multiple colors to recognize things. And the other thing is that, well, these images are great, but they are static. The cell is dead. So how can I see dynamics in that? And so we have been working very actively, and we are obviously not the other ones. It's an extremely active field of research, trying to get simultaneous multicolor imaging together with super resolution, and also trying to get dynamics information in there. And we actually succeeded that, and this was one of the images that I sent. It is for the advertising of this. So we managed to get 6 colors simultaneously and being able to label 6 different organelles simultaneously in cells by using an approach which I don't have time to explain to you. This is a technique that we implemented, and that essentially is implemented first in conventional confocal microscopy, fixed. What we decided it was to follow up on this frequency multiplexing approach to now being able to extend that into living cells. And what you see in here is a cell being labeled in which we are labeling microtubules, we're labeling lysosomes, and we are labeling mitochondria. And you see the amazing amount of mobility and interactions that now you could have by looking at this in real life using 3 simultaneous colors. And so you can see in that, and people are actually, and this is one of the research fields that we are following right now, is to map the interactome of cells. So how do different organelles and how different components interact in a spatial and temporal way? And you can see multiple interactions in here between many of these lysosomes and microtubules, and also with the mitochondria and the dynamics and the different dynamics that you see in there. And this is now a super resolution image in 2 different colors of, again, the mitochondria together with the microtubules that we obtained simultaneously using super resolution. So I think that now super resolution is getting into the hands of multicolor and also into the living cell world. Okay, so this is a lot of technical stuff. So for me, summary of this first part, there are an enormous amount of techniques now available, which are still challenges, which still have limitations, but essentially our eyes to the nano-world of living cells is now open, and many different groups, and my group, we are intensively collaborating with biologists in order to see, to visualize, but also quantify things that we did not have before, that we could not imagine to be able to do before. And so I'm just going to give you a few selected examples in there. One of the things that we did, so it comes a little bit darker on the screen in there, but what you're seeing in here is a dendritic cell, a living dendritic cells. These are cells of the immune system. The role of those immune cells, those dendritic cells, is to capture pathogens very efficiently. And in fact, the HIV virus is being captured by those dendritic cells, which are the ones which are very close to the mucose. And so how those dendritic cells, they capture and they internalize pathogens, and how does it do it with the HIV, and how does HIV escape the immune system? Because by escaping the immune system is when we develop AIDS, right? And so this was an experiment in which we use individual viruses, so each individual green spot that you see in here is an individual virus. And so we essentially expose the viruses to these dendritic cells, and you see that as the dendritic cells, they sort of move around, the virus lands on the dendritic cells, and they start to move in a very directional way to form a compartment. And by having this compartment in there, what the virus does is that hijacks the dendritic cells. It hides away from the degradation machinery of the cell, so that then when the dendritic cells travels to the lymph node, the virus that it was encapsulated in this compartment releases and is able to infect the T cells, okay? And so we were able, by using these techniques of single molecular dynamics, to be able to map the different stages of pathogen capture and infection to T cells. And this is essentially mediated by this receptor, which is on the cell surface of the dendritic cells, that capture the virus, and then upon capturing of the virus, the virus essentially changes and hijacks the entire cell machinery to do what it does. And I found this extraordinary, that the virus takes advantage of what the dendritic cell does and have in order to spread the infection. So we have been looking at these different receptors in there and trying to understand how does this receptor is able to capture the virus. And what we found out in here, and this is only combining this super resolution stat and this single molecule dynamics in which we followed the diffusion of individual receptors on the cell surface, to essentially we discovered that this receptor forms little nanoclusters. And the nanoclusters, they give essentially enough strength, because they are clusters, to bind to the virus. And then, therefore, the entire process starts. So it's the nanoclustering of these receptors, and these are little nanoclusters in the order of 50 nanometers that essentially are responsible for the binding and the capture of the HIV virus. And so essentially what we do, we use our techniques, we look, we visualize, we quantify it, and then we know the answers. Another example, which I think is extremely cool and extremely interesting, is to look at nuclear organization. So how is chromatin organized inside the nucleus? Okay? And so the textbooks that we all know is that essentially the chromatin inside the nucleus is essentially organized in this very hierarchical way, right? In which you have multiple rounds and rounds and rounds and wrapping in order to fit 2 meters of DNA in a 5 micrometer nucleus in there. And so this is the textbook idea in there, in which the helix of the DNA, it wraps around these histone proteins that then wrap around into these nucleosomes, helixes, and then they wrap around again into what it is called this chromatin fiber, and then we get the final chromosome in there. And so to actually look at this organization of this chromatin structure in intact cells, of course, it was just not possible. And if you would do a conventional microscopy image of the nucleus, this is essentially this very blurry structure that you see in there. And so if you now switch and you now use this super resolution storm microscopy, you start seeing very fascinating features in here in how the DNA and these nucleosomes, so this part of the wrapping of the DNA, occurs. And you can see that there is this bit on a string model. So you have pieces of DNA wraps around a nucleosome and another piece of DNA that wraps around the nucleosome. And essentially, we did not find the evidence for the formation of a chromatin fiber, which has been speculated in the field in that. So this is essentially a result that changes our view into how chromatin and how DNA is packed in the nucleus. And the other fascinating point in here is that when we looked at the nuclear, at the chromatin organization of different cells, stem cells, and fully differentiated cells, we saw a dramatic different organization of chromatin. And this is essentially representative super resolution images of what we see in the nucleus of the cell. So we see that in stem cells, the chromatin is extremely decompacted, you see. And when we look at fully differentiated cells, the chromatin is extremely compacted. And so we saw this difference in here. And so the level of compaction of chromatin also gives indication for the differentiation state of the cell in there. And this is actually, if you have seen this, then you're able to understand what the significance of it is. And so what the cells do is that on stem cells, as I told you, on stem cells, the chromatin is very decompacted, so you essentially have a lot of loosey parts and available parts of DNA available. So why is that? Because a stem cell has the potential. It needs to differentiate. It needs to change. And therefore, the DNA has to be more accessible. If the DNA is fully wrapped, it cannot be accessed. And so the cell essentially puts the DNA in a more accessible way so that the cell goes from a stem character to a more differentiated one. Once the cell is differentiated, it doesn't need to go into further changes. And then the chromatin compacts. And then it becomes very compacted so that there is very little DNA available for further changes in there. Okay, and the final example that I want to show you is some work that we're doing in collaboration with the Center of the Clinical Medicine in Oxford, and is towards this very fancy, very hot topic field nowadays, which is cancer immunotherapy. And actually the Nobel Prize in Medicine last year was given to cancer immunotherapy. And so it has been predicted already that immunotherapy, that is using the body to find cancer, is the solution towards cancer. And actually there are some clinical examples already of people that have been sort of exposed to immunotherapy process that the tumor cancer is just completely gone after 10 years of treatment. There are many drawbacks and there are many things that are not being understood nowadays. Still, so it's not a 100% success. But in a short summary, how does that work? You have your cancer cells in here, so these are the cancer, the tumor in there. And actually the tumor is something that is foreign to your body. And because it's foreign to your body, the immune system is ready, is prepared to kill anything that is foreign. So the immune system in principle has the capacity to kill cancer, but it needs to be trained in a specific way. And so what it was found out, and that's why the Nobel Prize came about, it was because there are these cells from the innate immune system, which are natural killer cells, that they are able to fight the cancer. But these cells, they need to be activated. They don't do the job on their own because they essentially control our autoimmunity, the basis, the more innate autoimmune system. And so they need to become activated, and they can become activated by these other dendritic cells that I told you about. Okay? And so many of the work in here is essentially trying to activate these cells in a very selective way to kill the cancer. And this has to be done by these dendritic cells in there. And so what we essentially did is that together with the group of Enzo Cerundolo, look at how do the dendritic cells have to activate these other ones in there. And we look at this particular receptor in there, and what we find out again is that in the way that this receptor organizes in space and in time, in nanoclusters, essentially defines the level of activation to these cells in there. And so I'm running out of time. I will just put you the conclusion in here. So essentially, this receptor that I told you about, it essentially forms very little nanoclusters, and that maintains these killer cells in a sort of a resting condition, okay? And so when the dendritic cells rearranges the actin cytoskeleton, what essentially does is that makes that this receptor forms larger clusters. And by forming larger clusters, is able to activate, to really trigger a massive activation of these natural killer cells, which are the ones that are going to attack cancer. Okay? And so this example of this very nice work, essentially what it tells you is that the degree of controlling in space and time different receptors essentially defines how much are you going to activate other cells to perform the job. In the same way that I told you about chromatin organization inside the nucleus in space and time controls the degree of cell differentiation. And I just want to conclude, and I think I'm coming very close to the message of Elena at the very beginning, it's only by combining multiple techniques and fields from physics, biology, chemistry that we're able to advance science. And I just want to cite this very nice quote that it says that progress in science depends on new techniques, new discoveries, and new ideas, and probably in that order. Making the techniques that we are making in our lab actually allows us to see new things that we never thought about it, and that actually generate new ideas for future research. And with that, I just want to leave the name of the people in my group. We work on 3 different topics, essentially in nanophotonics, the part of the nano antennas, a lot of single molecule biophysics, and trying to bring physics understanding into the biological processes, which I did not have any time to tell you about it today, and people working on immune receptor organization, and then a new project that I got from the ERC, which is in the field of nanomechanobiology. These are the collaborators and the funding agency. Thank you.
Muchas gracias por esta tan interesante y además muy didáctica, con lo cual ha sido
Gracias.
fácil seguirla. Y si tenéis alguna pregunta.
¿Cuál es el límite? ¿Habéis llegado a los 20 nanómetros? ¿Cuál es el límite? ¿Se rompe el límite de la diffraction? ¿Hasta dónde se puede llegar?
Se puede llegar muy lejos. Estas técnicas, de momento, y personalmente yo creo que será muy difícil conseguirlo todo, en el sentido de que tienes que hacer un trade-off. Podemos conseguir, de hecho, podemos conseguir una resolución equivalente hasta 1 nanómetro. Pero, ¿qué es lo que sacrificamos allí? Ese 1 nanómetro lo podemos conseguir porque podemos localizar moléculas con una precisión tremenda de 1 nanómetro. Pero esto significa que necesitamos utilizar marcadores fluorescentes muy estables y que nos den muchos fotones. Y estos marcadores no son compatibles con la biología. Entonces, han habido trabajos, pero esto no es compatible con la biología. ¿Vale? Podemos también intentar ir más rápido para poder tener dinámica. Si tenemos dinámica, sacrificamos en resolución. Entonces, tienes que hacer sacrificios o compromisos. Entonces, dependiendo de lo que quieras, en el futuro dependemos y seguimos dependiendo mucho de los químicos orgánicos, de los que sintetizan, como estas técnicas están tan basadas en las propiedades fluorescentes, y de hecho hoy en día la cantidad de empresas que han nacido y que se dedican justamente a mejor síntesis de moléculas orgánicas para que nos den más fluorescencia, para que vivan más y de manera de poder aumentar esa resolución equivalente.
María, no sé muy bien cómo hacer la pregunta, pero el contexto sería la interfase entre las imágenes de superresolución que obtenéis
sí.
con la información que procede, por ejemplo, de información estructural que hemos obtenido pues, los espectroscopistas, cristalógrafos, o sea,
sí.
información estructural a muy alta resolución.
Sí.
¿Vale? Entonces, ¿cómo se complementa una cosa con la otra? En el sentido de, por ejemplo, el ejemplo que has puesto de la inmunología en el cáncer.
Sí.
¿Se interpreta vuestras imágenes con información que se conocen a nivel molecular?
Muchísimo.
¿O las imágenes de superresolución aportan una información completamente novedosa que hacen reinterpretar la información molecular?
Las dos cosas.
No sé si me he explicado.
Sí, te has explicado vale. Perfectamente. Las dos cosas, y de hecho hoy en día hay varios grupos que intentan hacer biología estructural con estas técnicas de resolución. Entonces, claro, hay casos, y esto obviamente lo sabes tú muchísimo mejor que yo, en donde es muy difícil o imposible cristalizar una proteína. La cristalización es complicada, está toda esta nueva técnica que de hecho ganó el Nobel Prize el año pasado de cryo-EM para poder resolver estas estructuras, y las técnicas de superresolución están allí, haciendo básicamente. Entonces puedes ver inclusive charlas de varios grupos. Jonas Rees, por ejemplo, en el IMBL, él está complementando información estructural con superresolución e intentando hacer matching de los dos tipos de estructuras. Entonces, trae información complementaria. ¿Cuál es la ventaja de hacerlo con estas técnicas de superresolución y la fluorescencia? Que tienes más parámetros con los que puedes jugar. O sea, tú puedes hacer structural biology in the cell nowadays. ¿Vale? Que esto es completamente impensable, ¿no? En otro tipo de bueno, cristalografía, por ejemplo, o esto. Entonces, es esto hasta allí, hasta donde se llega. Otro tipo de avances que se están haciendo hoy en día es complementar diferentes técnicas, utilizar la microscopía de fluorescencia en la misma muestra, Entonces, haces como múltiples la misma muestra, miras con la microscopía convencional, luego te vas a microscopía de superresolución para tener la escala nanométrica, y luego esta misma muestra te la llevas a cryo-EM para tener información estructural. Y luego combinas todas estas escalas, que esto es, se está haciendo, pero esto es un gran challenge, que además, o sea, obtener una imagen es un año, una imagen, con información 3D. Imperial College hay todo un departamento enfocado a esto. Sí.
Verás, yo, bueno, tengo dos preguntas, dos o tres. La primera es acerca de la Near Field Scanning Imaging. Si tú tienes 20 nanómetros de precisión en la antena, necesitas un barrido también con una precisión de 20 nanómetros. ¿Cómo se hace ese barrido?
Esto vas haciéndolo, básicamente lo que vas es píxel per píxel cuando vas haciendo la imagen allí. Entonces, lo que ocurre es que imagínate tú que tienes un spot de este tamaño que quieres hacer la imagen, pues vas con tu antena y básicamente pues vas haciendo un escáner píxel por píxel por encima de cada una, como lo harías con una microscopía confocal.
Te quiero decir, ¿qué tipo de tecnología tiene esa precisión de mover una cosa de 20 nanómetros con una precisión de 20 nanómetros, digamos?
Sí.
¿Es con piezoeléctricos?
Sí, claro, utilizas piezoeléctricos. De hecho, o sea, tenemos una resolución de unos 20 nanómetros, pero utilizamos elementos para irlo moviendo. Y la precisión que tenemos para irlo moviendo es de 1 nanómetro. O sea, cada un nanómetro vamos haciendo el mapping.
Perfecto.
Vale.
Y ahora, en las imágenes con movimiento, digamos, ¿qué tipo de tecnología es? Entonces, ¿es la de fluorescencia confocal estándar?
No, la técnica se llama EPI, es EPI Illumination, no utilizas confocal. Puedes hacerlo confocal, pero es muy lento porque confocal requiere el escaneo, o sea, o escaneas el láser o escaneas la muestra. Esto es lento.
Claro,
entonces se utiliza otra técnica que es simplemente, pues iluminas toda la muestra y la fluorescencia que la recojas te la llevas a una cámara, un CCD cámara. Sí.
Y entonces, es de nuevo coger fluorescencia, digamos, de las... y mi pregunta es relacionada con esto, que creo que la has más o menos respondido en la anterior pregunta. Para ver la fluorescencia, digamos, ¿hay que añadir moléculas extra?
Sí.
Sí, ¿no?
Sí, que es la gran desventaja de la microscopía basada en la fluorescencia. Tienes que marcar.
Hay que
utilizar marcadores fluorescentes. Ajá. Y esto, pues hay muchos grupos de investigación que intentan implementar otro tipo de técnicas que son label free, pero que tienen muchas limitaciones, que son muy potentes, que son label free, tienen la gran ventaja, pero tienen muchas limitaciones, sobre todo cuando las quieres aplicar a la variedad y a la complejidad de la biología, ¿no? Es por esto también que los biólogos pues continúan, o sea, su primer tool de batalla es la fluorescencia, porque te da toda esta versatilidad, pero el precio a pagar es que tienes que marcar. Sí. Hola.
Muchísimas gracias. A mí me gustaría saber tu opinión sobre la combinación de estas técnicas que nos has presentado, increíbles, con la posibilidad de hacer espectroscopía en el microscopio, o sea, hacer imágenes espectroscópicas y obtener esa resolución que nos estás mostrando a través de la espectroscopía.
Ya.
Y también, o sea, es que me doy cuenta de que son dos mundos que deberían de estar más unidos y no sé por qué no se juntan, o sea, son como conjuntos disjuntos.
Sí.
Los que hacen el abordaje de un lado y los del otro, no sé, si entiendes mi pregunta.
Sí, claro que claro que la entiendo. ¿Existen algún tipo de intentos? Tú conoces muy bien este campo, básicamente lo que yo he explicado. Por ejemplo, en el caso de estas superresoluciones basadas en single molecule detection, básicamente lo que estás haciendo allí es que estás básicamente midiendo la cantidad de fotones que recoges por una molécula individual y usas la intensidad para asignar una posición. Obviamente, esos fotones que estás recogiendo también contienen información espectroscópica, contienen información de wavelength, contienen información de orientación. Se puede hacer, y lo puedes hacer, y hay algún que otro paper en donde se ha demostrado, por ejemplo, pero significa añadir, supongo, y mi opinión aquí, es que significa añadir complejidad a un equipo que es básicamente trivial. Es que un equipo de superresolución es una chorrada. O sea, es simplemente un objetivo, un láser, dos láseres, uno para hacer switch off de las moléculas, uno para hacer switch on, un objetivo y una cámara. Más nada. O sea, el Nobel Prize que le dieron a Eric Betsic estaba desempleado. Esta es una anécdota que además él siempre cuenta. Él estaba desempleado y con los restos que él tenía de una cámara, de un objetivo, lo montó encima de una caja en el salón de su casa. Y tardaron 8 horas en generar una imagen, pero generaron una imagen de superresolución. Entonces, es trivial. Claro, si tú añades información espectroscópica, de alguna manera va a pasar dos cosas. Lo primero, que tienes que enviar parte de los fotones a otra cámara, con lo cual pierdes fotones para hacer la imagen. ¿Vale? Porque necesitarías una cámara para recoger intensidad y hacer la imagen de superresolución y otra cámara, por ejemplo, para dispersar la wavelength y poner el espectro. Para poder, claro, tienes que al menos separar 50-50%. ¿Vale? Con lo cual, en la imagen de superresolución pierdes, así a ojo, un factor de 2 en la resolución espacial. Y luego aquí tienes que integrar por mucho más tiempo para poder recoger el espectro de una molécula. Entonces, por ahí van los tiros.
Bueno, me estaba refiriendo más desde el punto de vista, no hacerlo a la vez simultáneo, que lo comprendo, sino hacer un abordaje, ya sea, aunque fuera solo en los inicios, luego para entender mejor esas imágenes y para interpretarlas mejor, en el que tú tuvieras la posibilidad de hacer las dos cosas a la vez, y entonces eso serviría de puente y daría muchísima más información en el diseño de alta resolución, en mi opinión, de modesta.
Vale.
Nada más, es que me extraña que no se llegue por ahí.
Bueno,
no
pero es que lo tienes que hacer, sí, te entiendo. Hay algún que otro grupo que lo está haciendo, ¿eh? Y de hecho midiendo, por ejemplo, Fluorescence Lifetime Imaging, haciéndolo en regiones pequeñas. Claro, hay limitaciones. Fluorescence Lifetime Imaging necesitas un detector muy rápido, entonces necesitas acoplarlo con una PD, con lo cual tiene. Y claro, lo tienes que hacer casi que simultáneamente, porque recuerda que te tienes que estar switchando las moléculas individualmente. Si lo haces al final, te has cargado toda la fluorescencia, con lo cual no tienes nada para generar espectroscópicamente. Sí.
¿Alguna pregunta más?
Bueno, yo desde fuera del campo me ha parecido tremendamente interesante y tengo la curiosidad de ¿se aplican este tipo de técnicas en la parte dinámica con superresolución a la actividad neuronal, a registrar?
Muchísimo. Sí.
Lo sospechaba, pero me ha parecido curioso que no pusieras... Bueno, claro, hay que elegir ejemplos.
Porque yo soy física, te digo la razón. Porque yo soy física, porque la biología es terriblemente complicada para mí, y cuando empecé a hacer cosas de biología por casualidad, las comencé a hacer con inmunólogos, que les fascinaron estas técnicas y tal. Me lo han propuesto algunos investigadores que trabajan en la parte de neuronas, pero tendría que aprender demasiado, que es demasiado, es muy complicada. La biología es demasiado complicada, entonces yo me quedo en mi mundito. Pero sí que hay un gran interés y un gran avance y un gran potencial, por ejemplo, y de hecho hay muchísimos trabajos para resolver lo que se llama la sinapsis neurológica, mirando a escala nanométrica en esto. De hecho, el grupo de Melike Lakaramiali en UPenn es uno de las especialistas trabajando en esto.
Muchas gracias.
Sí. Gracias a vosotros.
Y deciros que se acaban los ciclos de esta temporada, pero que ya estamos trabajando para la siguiente, que os esperamos a todos y que esperemos que sean tan fructíferos y que podamos conseguir las 5 IES que tan amablemente nos habéis presentado en la exposición que ha hecho la vicepresidenta. Pues muchas gracias. Hasta septiembre.
Gracias. Muy bien.